No, I'm psychology. Yeah. Yep. I'm just here for the colloquium.
So we're going to do a little bit something different as our introduction. So Ruben, you can take it away.
David is a Southern Ontario boy. He went to school at the University of Toronto, Mississauga, where he already gained significant research experience and even authored a very nice paper. Afterwards, David went to Wilfrid where he did his master's and developed refined expertise at working with food flies. His master was quite prolific. He produced 3 very good publications, and then he came to my lab about 3.5 years ago with very clear ideas of what he wanted to do for his PhD, which led to him earning an NSERC fellowship. David has been quite prolific. I produced 2 top publications already. And is busy producing at least 3 more because he plans to graduate in 4 years, meaning this summer. David writes well, and as you all know, he also presents well orally, and you'll see it if you haven't seen him present before. So I look forward to watch his talk, and with this, I will let David switch to his title for today. So you can take it, David.
Thank you, Ruben. Bye.
All right. Okay, so this mic is not good now. Hopefully we don't get the feedback. All right, so thank you everyone for coming. Today I'm going to talk about what I'm going to try to cram my 4-year PhD dissertation into one 40 to 50-minute talk about the causes and consequences of variation in reproductive behavior. Another glitch, and that's because this isn't plugged in. All right, so throughout all kingdoms of life, we see considerable individual variation. And for evolutionary biologists, the solution to this is quite intuitive. So the planet's really big, there's lots of diversity in ecosystems, so we would expect that natural selection would result in the diversity of life due to different adaptations in different ecosystems. However, we also see considerable individual variation within species as well. So 2 very familiar examples of this are us and our best friends, dogs. I actually shouldn't generalize there because my father, who is here, does not like dogs, and you should all publicly shame him for that. So when we think about individual variation within species, this isn't as intuitive as to why we might see so much variation. And from an evolutionary perspective, we can make the prediction that in a given environment that is static, we would expect all individuals to eventually conform over many, many generations to fixate for the same phenotypic traits. But of course, we know this isn't the case. So one solution to this, or one of the many solutions as to why we see so much individual variation, is phenotypic plasticity. So I'm talking about plasticity here strictly from the biological perspective, which is the idea that a single individual or a single genotype more broadly is able to express multiple phenotypes depending on environmental influences. So here's an example quantitatively of no phenotypic variation. So each line on this figure here represents a different individual, and we can see here that over an environmental gradient on the x-axis, the magnitude of whatever trait we're measuring isn't changing. So there's no plasticity in this specific case. We also have cases where individuals might conform to change in the same magnitude and in the same direction. And this is an example of phenotypic plasticity. And we can see in this case, all 3 individuals are actually increasing at the same rate. So although there's phenotypic plasticity, we can say that there's no variation within this plasticity. And then a final case is when we have individuals changing their traits or changing their phenotype over an environmental gradient, but in different ways. So here we can see the red line is growing, if we're considering size, is growing bigger. The blue line is growing bigger, but the black line is growing smaller. So in this case, there's actually genetic variation in phenotypic plasticity, which is of course, important if we're going to look at its evolution. So one of the environmental factors that might be really dynamic that allow organisms to modulate their traits and behaviors is the social environment. So as you can imagine, many different social factors, such as competition or the sex ratio in the population, are going to influence organisms' behaviors in ways that might benefit them more in that particular circumstance. So for example, if we consider two genetically identical individuals, an individual in an environment with low competition might benefit from a very different behavior or set of behaviors compared to an individual in a high competition environment. And I'll go into a bit more specific examples of what these behaviors might be in a little bit. But first, before we do that, we have to consider when it comes to studying sex and sexual traits, which of course are very important from an evolutionary perspective, because in order for evolution to successfully occur in a sexually reproducing species, sex needs to occur. So organisms maximize their reproductive success through two broad categories, the first being pre-copulatory strategies, which improve an individual's success before copulation or before sex occurs. So things like how attractive or sexy that individual is, how much effort they actually put into finding and acquiring mates, and of course things like competitive and contests to gain access to mates. But in species that mate multiply, which is extremely common throughout the animal kingdom, individuals also have to worry about their success after copulation occurs, and these are post-copulatory traits. So these are traits that might enhance an individual's ability in sperm competition. So their ability to outcompete the sperm of rivals and the reproductive tract of females in order to fertilize eggs. And females are actually able to discriminate between different sperm and decide what fertilizes their egg, and this is called cryptic mate choice. Males have evolved accessory proteins that are transferred in the ejaculate with the function of enhancing success, enhancing fertilization, enhancing sperm competition. And then, of course, any offspring investment that occurs after would fall into post-copulatory as well. So given all these different traits that individuals have to focus on to be successful in reproduction and thus being successful in passing on their genes, you might imagine that no one individual would be capable of being the best at every single aspect of reproduction. And this can be explained nicely by life history trade-off theory, which is essentially the idea that in natural environments, there are limited resources, and therefore the amount of resources an individual can acquire is going to be limited. And due to this limitation, individuals have to make decisions whether or not they want to invest into things like their growth or their overall maintenance and survival, all the way up to their reproductive tactics, where individuals have to decide, or their physiology has to decide for them, what reproductive traits are the most beneficial to invest in given their current environment. So sometimes these trade-offs are constrained completely by genetic factors. So here's a paper from my master's, or a figure from a paper from my master's degree, where I looked at male success in a uniform environment. So the only thing varying here is the genetics of the individuals. And each dot on this plot represents a single male genotype or genetic background. And on the x-axis, we have a measure of fertilization success, so post-copulatory. And on the y-axis, we have a measure of success in mating and the presence of competition, so a pre-copulatory strategy. And we see here this nice negative correlation where male backgrounds that are really good, or relatively in this sample, really good at fertilizing eggs, aren't as good when it comes to male-male competitions. And sometimes, of course, the environment can be responsible for these constraints and decisions as well. So here's a nice example I found, missing the reference, but I can get that for you, where they studied guppies and took guppies that were either raised in a clear water environment or a turbid, so basically a cloudy water environment. and found that the guppies raised in the turbid environments were exhibiting significantly more reproductive behaviors. And from an evolutionary perspective, the idea here, what the authors propose, is that given that the visual ability of these guppies is reduced in the turbid environments, it makes sense for them to invest much more into reproductive traits when they see a mate, because it might be much harder to find a mate when the water isn't clear. So based on these examples and the literature at large, it's relatively clear that there is strong evidence that individuals within many species are really, really good at strategically investing into different strategies depending on their environment and, of course, their genetics as well. However, we might all be familiar with the fact that sex is not only a one-player game. There's always going to be two individuals involved, a male and a female in most species. So therefore, we have to consider that the variation in reproductive strategies not only has the consequence for the individual that is expressing the plasticity, but we also have to consider the consequences on the individual that is being mated with or attempted to be mated with. But before we consider this question, we have to consider some aspects about how the sexes interact in nature. And an important principle to consider is that of anisogamy, which is the concept that in a majority of sexually reproducing species, there's an asymmetry between males and females when it comes to how much they invest into reproduction. So typically, females are going to be investing a lot more metabolically into creating eggs, and also post-reproduction in offspring rearing. And typically, males are going to do less with sperm and less parental care, but that varies immensely between species. And as a result of this, sexual dimorphisms or differences between the species evolve to help individuals meet the selective pressures of these differences. And two examples of these are the optimal mating rate, where males are expected to have a much higher optimal mating rate, and males are, as a consequence, also expected to compete much more intensely, because if there's a higher optimal mating rate, more males are going to want to mate, and therefore there's going to be more competition to mate. So what this can be defined as is an example of sexual conflict, which is broadly the idea that males and females in many species have different strategies for optimizing their success. And this has led to a process known as sexually antagonistic coevolution in nature, whereby males have evolved a variety of exploitative traits that attempt to exploit this low optimal mating rate, this choosiness aspect of female mating behavior, through mechanisms such as potentially sexual harassment, or in more extreme cases, forced copulation. And of course, from an evolutionary perspective, this is going to increase the fitness of the males because they're having more reproductive opportunities, but come at a cost to the females that they're mating with because they're exploitative traits. So now that females are met with this selective pressure, we can make the prediction, and we've seen in examples as well, that females will evolve traits that attempt to resist these exploitative male traits through things like choosy mate choice behavior and discriminating with what males are the best to mate with. And of course, this is going to now increase female fitness, but at a cost to males. So the grand, idea or synthesis of my dissertation was to bring together these two relatively well-understood areas of research of sexual conflict and plasticity to form the logical statement that if individuals who are modulating their success through plasticity for their own individual benefit, which is well understood, and we also understand very well that the reproductive interests of sexes often conflict, including in the fruit fly, which I'll talk about in a minute, then we can make the prediction that plasticity in the reproductive behavior of one sex is going to have significant consequences for the fitness outcomes of the opposite sex, or in short, sexual conflict. So the broad objective of my dissertation was to study plasticity, how it occurs, why it occurs, and use these differences in reproductive behaviors to quantify the fitness effects of plasticity on individuals as well as their mates. So I did this by breaking my thesis into 4 general predictions. The first, that social experience will affect male behavior and then thus his own reproductive success. Then that social experience will affect male behavior, but now the sexual conflict aspect, the reproductive success of the females that they're mating with. And then I wanted to look at the female side of things, which is the social experience affecting female behavior and the success of their mates, and then social experience on female behavior and success of their own selves. So I know here social experiences it's a very vague, broad term. Bear with it for now. Once I go into each experiment, I'll go into detail. But today I'm only going to talk about 1 to 3, partially because of time and partially because of completion. So before I do that, I want to emphasize, as many people know, what I think about the fruit fly as a powerful model. Before I do that, I want to apologize if this picture offends anyone, because in my course reviews last semester, I had not 1, but 2 people write in the need for improvement area, please stop using so many pictures of fruit flies having sex in your slides. So apparently I'm just desensitized to it, but I mean, if that's their only criticism for me as a teacher, I shouldn't complain, but just a warning there. So fruit flies, of course, are a classic model in a lot of different disciplines in biology, specifically in studying sexual conflict and plasticity. So you can imagine if I'm attempting to synthesize these 2 areas, they would be ideal. As well as that, it's very simple relatively simple, to control their environmental and their life history, and we do that by keeping them in very controlled conditions. So we know that any variation is due to something we manipulated within its lifetime, or most of it. And then, of course, there are also a variety of genetic techniques we can use to generate clones very easily, which allow us to control whether or not differences are due to the environment we're manipulating or due to the genetic background of individuals. And in our lab, we use a genetic dihybrid approach. So this is essentially taking an individual that is a full clone and then crossing it to another individual that is also a full clone, but with different genes. And this gives us these dihybrid individuals because half of their genotype or one of their haplotypes is from the clonal mother and one's from the clonal father. So essentially what this does is it gives us full clones, but since they're now hybrids, we don't have to worry about the deleterious effects of inbreeding and having the exact same genes on both backgrounds. So we can do this with as many different backgrounds as we want to produce a lot of different clone lines. So now I'm going to talk about my first project, which was male experience and male success. So as I already alluded to, males compete very intensely in a lot of species to gain access to mates. And the outcome of these competitions, and more generally just regular competitive competitions over things like resources, or even in humans, sports and games, animals exhibit very specific behavioral responses to the outcomes of these contests, known as winner-loser effects, where winners are much more likely to win a subsequent conflict, and losers are much more likely to lose a subsequent conflict. So here's just an interesting study looking at some historical data in tennis matches, and we can see that in general, individuals, or in both males and females, that score more than half of the points on the previous set, meaning they won that set, have this big increase in their odds of winning the next set. So the behavioral aspects of winner-loser effects are quite well understood, but the response on, or the effect on mating behavior and reproductive success isn't. So I wanted to ask, what are the consequences of winner-loser effects on male reproductive success? And more specifically, do losers fare better in any of these different domains of mating, so considering pre- and post-copulatory traits? So to do this, I first had to generate a methodology to experimentally generate winners and losers. And I did this by using this system we brought up in our lab, essentially, where we took flies that were artificially selected to be hyper-aggressive. So they're by nature very aggressive. And we found that within these flies, males that are 5 days old are way, way more aggressive, around 18 times here on average, than males that are only a day old. So we have these really aggressive males and these really docile males. And using these males, we can pit them against focal males, and we can see here that on average, when a focal male faces a young competitor, they are the ones being the aggressor, and when facing the 5-day-old male, they're the ones receiving the aggression. So we're essentially using this method to rig the fights to generate losers or winners and losers respectively. So the first prediction of this study was that compared to losers, winners will have greater success in the presence of male-male competition. And I did this by placing a single male from one of 28 of the different dihybrid backgrounds I was talking about earlier, and paired him with either a hyper-aggressive male or with a docile male. After this 4-hour experience phase, we removed the initial competitor, so that hyper-aggressive or hyper-docile male, added a new competitor to our focal male, and then added a female to these two males together. And then right away, these males will start trying to compete to mate with this female. And we scored as our dependent variable the mating success of our focal male, whether or not he was the one successful in mating or not. So it's just a binary outcome, win or loss. Yeah. No, so sorry if this wasn't clear. The focal males are all 4 days old, and then we're only manipulating the age of the competitor to manipulate aggression. Does that make sense? Okay, good. Okay, so what we found was, as we predicted, the winners that won the previous fight outcompete a new competitor at a greater frequency compared to losers. So first looking at these boxes, which each contain the different dihybrid averages for mating success, that on average winners are outcompeting the second mate more frequently. And then if we look at the genetic variation in this effect, so here now at this reaction norm plot, each line here represents a single dihybrid genotype we tested. We can see that there's also, although it's not significant, there's some qualitative variation here. So we can see that some lines are getting much better when they're a winner, as we predict. Some lines exhibit not a lot of plasticity, and some lines actually have this odd opposite effect where they do worse as a winner. But it's not significant, but the variance component is relatively high, so it's something to consider. All right, so next we wanted to test another aspect of precopulatory success, which was female acceptance. And we did this by generating winners and losers in the exact same manner. But now, instead of introducing a new competitor after we remove, so that's the first competitor, and now instead of removing or introducing a new one, we just introduced the female. So what's going to happen now is we scored the dependent variable as the amount of time it takes for that male to successfully court that female to mate with him. And once again, we do find the same effect, or the effect that we predicted, whereby winners are mating faster, meaning they're doing better in this test. So you can see that clearly on our boxes. And then once again, we see a lot of crossing over between our different genotypes. So visually, there's some variation, but it's not significant. So next, we wanted to test the effect on post-copulatory success. So things like sperm competition, and we predicted that losers in this case will stimulate more offspring production and will father more offspring when they're the first male to mate. So the rationale here is that there's a growing body of literature to suggest that one way losers might compensate in the fact that they might not have a lot of mating opportunities is to invest as much as they can into the mating opportunities that they do acquire. So the experience phase here was exactly the same, so I'll just brush over it fast. The only difference here is we tested 6 lines. Since we didn't have enough power to detect genetic variation with all of our 28, we just reduced our lines. So in this test, we introduced the focal winner or loser from the experience phase to a female once again, but here the female has a brown eye phenotype. And this brown eye phenotype is controlled by a recessive gene, which means that you need 2 copies of it to be expressed. So that means that this wild type male that has the red eyes, you can see it's very subtle, the color of the eyes, but any offspring that he fathers will have red eyes because red is dominant to brown on this gene. We then took the female and put her in a vial and let her lay her eggs overnight. And then 2 weeks later, we counted those eggs, and that will tell us how many offspring that male was able to stimulate for that female to lay. Then we took the female from the previous day, so the slide I just showed you, and put her in an arena with a new competitor, but this male competitor has brown eyes. So that means any of her eggs that are fertilized by that male sperm will have brown eyes because she'll have 2 copies, or her offspring will have 2 copies of the brown eye allele. And by doing this, we were able to let her lay her eggs, and then 2 weeks later, when we counted the offspring, quantify the amount of paternity that the first male, that was the winner or loser, was able to defend in a competitive event based on the proportion of offspring sired by that male. And as we predicted, we found that losers stimulate greater offspring production in their mates. So here we can see on the x-axis, the losers produce, on average, slightly more offspring after a single mating, but you can see the distributions are very different, even though the means or the medians actually are relatively close. And then looking at the genetic variation, once again, we just simply don't have the power to detect anything, and there's no significant genetic variation. So now looking at the ability to defend paternity, we find the same positive effect, whereby losers, once again, are outcompeting the winners by defending a higher share of paternity when a second male mates with a female they had already mated with. And then once again, we see no genetic variation. So this brings us back to the initial question of this study, which was, can losers still be winners? So basically, do losers have other mechanisms of improving their reproductive success other than what we would generally imagine in nature as being the ability to acquire mates? And as our data shows, yes, it actually does appear that winners, even though they have the higher pre-copulatory success, it's the losers that are doing better in post-copulatory arenas. And as I mentioned when I was outlining my prediction, this is expected because if an individual is getting less mating opportunities, it makes sense to invest as much as they can in the mating opportunities they do get from a trade-off perspective. So the plasticity, however, is relatively weak across genotypes, meaning that all individuals relatively respond the same way to this winner-loser effect, and thus allowing us to form evolutionary hypotheses about these effects because we know more about the sexual selection involved. All right, so now we'll move on to part 2, where I'll talk about male experience, but now looking or focusing on the fitness of his mates. So as I already mentioned, sperm competition is a very important aspect of competition, especially in fruit flies, whereby males not only have to compete before sex, but after sex as well. And as it turns out, sperm, or the amount of effort a male invests into sperm competition, not only is affected by winner-loser effects, but also by the amount of competition in their perceived environment. So here we can see that males that were reared in an environment with multiple competitors, in this specific case, I think they had 3 competitors, and males that were reared in an environment with no competitors, the males that had competitors are transferring much more or significantly more sex peptide, which is one of the accessory proteins I mentioned that help males succeed in sperm competition. This makes perfect sense as well, similar to the winner-loser effect aspect, because males that perceive high competition might perceive the risk of another male stealing their paternity, so therefore it makes sense to invest in the mechanisms that help defend that paternity. So linking this to sexual conflict, we know that the competition against males, which has led to the evolution of these accessory gland proteins, is actually harmful to females. And we also know that a high optimal mating rate has led to the evolution of behavioral aggression, like sexual harassment, which is also, of course, going to be harmful for females as well. So linking these two together, I predicted that if traits that are enhancing sperm competition are harmful for females, as we know very well, and that males that are investing more into these traits because they're housed with rivals, then we would expect that these males housed with rivals are going to be more harmful or have a larger net negative effect on the fitness of females in both longevity, lifespan, and reproduction. So that's basically my prediction, that I would predict females that mated to males housed with rivals will die younger and produce less offspring. So to do this, I first generated a male experience phase to manipulate the perception of sperm competition. And males were either housed alone in a vial or they were housed with 2 competitors to generate that low or high risk of sperm competition. And they were housed in these treatments for 3 days. Following this 3-day exposure period, these males were now introduced to a single female who has not mated in the past and put in a vial with her for 3 hours. Within these 3 hours, they'll typically always mate. Then the male was removed. The female was allowed to lay her eggs. She was moved to a new vial 2 days later, and then introduced to a new male from the exact same treatment as she originally was, 10 times over 20 days. So in other words, every female in our experiment was either exposed to a competitor experienced male 10 times or a no competitor experienced male 10 times over a period of 20 days. So the dependent variables we quantified were first longevity. So this is quite self-explanatory. We just simply checked for female survival every morning, during, and after the experiment. But we also quantified the amount of offspring, or the reproductive ability of these females over her lifetime. And we did this by looking at the vials that were collected in each of the 10 days where males were exposed to females, and kept the eggs and let them incubate for 2 weeks, in which we were then able to count the number of offspring that each female produced over the course of these 10 days of her life. And what we found was, when looking at longevity, as we predicted, the females repeatedly mated to males housed with rivals do indeed die younger, slightly younger on average. So looking at these survival curves, which basically plots the number, or the proportion actually, of females that are alive within each treatment over time, we can see that the curve where females were mated to competitor males is lagging slightly behind the no competitor curve, meaning they're dying slightly younger on average. So we actually replicated this experiment in 2 different populations to test if this effect was general or if this was just a one-off thing we found. And we did indeed find the same effect in a separate population, whereby the females mated to competitor males are dying slightly younger on average. So the next thing we wanted to look at was how these different male treatments affected female reproduction. And here we found, firstly, when looking at total life reproduction, contrary to our prediction, there was no difference between the treatments. So looking just at the effect of male treatment, there was no difference in the total lifetime output. However, we did find a really interesting effect in the interaction between treatment and day. So in other words, the way females respond in their change in production over time, depending on what treatment they're in. And this is characterized by the pattern whereby the females that were mated to these competitor males actually produce more offspring, or have higher fitness in this context, early in life, but later in life this crossover occurs, and we can see now they're actually performing at a disadvantage compared to the no competitor males. Looking at the other population, we see the same effect. So at first you might see the offspring curve is quite different. That has to do with the type of population, which I don't have time to explain now, but if you want to ask me after the talk, I'd be happy to explain that. But the important thing here is we see that same interactive effect, where even in this population, the females that were mated to the competitor males do better in terms of offspring production in the first few days of their life, and then right around this point, day 8, they start to do worse consistently over time compared to the females that were mated to the no competitor males. So when we submitted this to a journal for the first time, my story, or my interpretation of that data, was essentially that we see that the females mated to the competitor males are on average dying younger. And although the offspring production isn't decreased overall, it's being affected. So therefore, we can conclude that overall, they do have decreased fitness because they're dying younger. And the reviewers weren't satisfied with that argument, because they claim that early life production in a species like Drosophila, which has a really typically an increasing population growth rate, matters potentially a lot more than production that happens later in life. And we saw in my offspring curves that it was actually the females that were mated to competitor males that were doing better early in life. So to absolve this, we performed an additional analysis where we looked at a metric of fitness called population growth rate. And this is essentially a metric that will take into account not only the survival or the longevity of each individual, but also the offspring they produce over life, with a higher weighting given towards the offspring that were produced early in life. And what we can see here is that there's actually, when we take both factors into account, no difference between treatments in population growth rate. So in other words, with this metric of fitness, we don't see any differences. But what we do see is if we plot our two metric of fitnesses with each other, so population growth rate, and lifespan, this really interesting negative correlation that really fits our trade-off hypothesis, whereby males seem to be causing females, the females that are living really long after being mated to these males are also the ones that have lower population growth rate. So that means that the females that are dying really young are producing a lot of offspring early in life, which means that these two different reproductive strategies, early life reproduction and survival, might be trading off with one another. And this trade-off is modulated by the male experience. So to summarize that, can we conclude that competitive males are more harmful? Well, the context of the environment certainly influences the plasticity of the males, but it's difficult for us to conclude that this is having an actual difference on the fitness of females that they're mating with. But with that being said, the results still show that there's a significant effect on how males manipulating different life history strategies of females, whether or not they invest early or late into reproduction, and whether they live shorter or longer lives, which is still very interesting. So the final part I'll talk about, I should have time, is female experience, and now looking at its effect on male success. So before I'm talking about manipulating a male social environment and looking at those fitness effects, now we're going to manipulate female social experience and look at the effect on her mates. So in the previous study, we showed that males can use their cues to affect the mating and reproductive traits of females. So by logic, we would want to make the same prediction on the opposite end of things and say that female social experience should also be able to do the same in modulating traits that resist male harm or resist male manipulation. So here's a little bit of details about sexual conflict on the female side of things. So the resistance aspect. So the intense pressure to mate through this increased male optimal mating rate has led to the evolution of choosiness, where females are very careful to discriminate on which males might be harmful for them or not. And they also express mechanisms that degrade the toxic side effects of the accessory gland proteins that are transferred in the male ejaculate. So the prediction I made was that males that are courting and trying to mate with females who have experienced a high level of sexual harassment or harm in the past will have lower reproductive success compared to males that court with females who have experienced low harassment. The idea here being that the high harassment should, or might predict, cause plasticity in the female behavior to help her reduce the effects of coercive mating in the future. So to do this, we first generated two unique female experiences, which were a low harassment and a high harassment trial, where the low harassment essentially contained a single male, and the high harassment treatment contained 3 males. So the females were placed within those treatments for 2 days. And within that 2-day period, we photographed these trials every 2 minutes. So this allowed us to go back and look at these photographs and actually count the number of matings that occur within each treatment, so we can later on determine, is there actually a difference in the amount of harassment that the females are experiencing within our treatments. The females were then placed in a vial alone. And then following this period, we tested for male success. So this first part of the simulation, this is what I showed on the last slide. So after the females are in that vial, a day later, they were placed into a vial with a new male, and we scored the success of those males by measuring mating latency and mating success. And also, you can't really see because it's overlapping there, but these males have that brown eye phenotype. But in this experiment, it's a little different because these flies, the brown eye phenotype is actually controlled by a dominant allele. So that means any offspring produced by this male will have brown eyes, and any offspring produced by these original competitors will have red eyes, allowing us to gain an idea of paternity success as well. We removed those males after the mating occurred and allowed the female to lay her eggs overnight. 2 weeks later, when those offspring became adults, we identified the paternity and counted the number of offspring produced. So first, we wanted, as I mentioned, to look if there were actually differences between our two experience treatments. It probably would have actually made sense to show this figure before the previous slide. So that's fine, though. This is basically showing what's going on within that 48-hour period of high harm and low harm exposure. And we can see that on average, females are mating more as we would predict because there's going to be more males that want to mate, slightly more on average compared to the one treatment, which is reassuring because that means that there is a quantifiable difference of experience going on between our two treatments, and we're not just saying there's 3 males in one and 1 male in the other. And as we predicted here, that the females that experienced high harassment do indeed take longer to mate with a second male, meaning that the second males have lower fitness when trying to court females that experienced high harassment. And we can see that here on these survival curves where over time, the females that are being courted who experienced high harassment in the past are taking longer on average to mate. And at the end of the trial, which lasted 14,000 seconds, which I think is 3, maybe 4 hours, the total number of females that mated at all was lower compared to the females that experienced low harassment in the past. So there's clearly this effect whereby the previous experience is affecting the success of the next male. So the second thing we wanted to look at was the paternity success as well, because there might be stuff going on post-copulatory wise as well. And we do indeed see an effect here as well, where the males that had the highest paternity share tended to be from the treatment of females that experienced low harassment. And this could simply just be due to the fact that the females in the high harassment treatment that experienced more matings, there's more sperm competition going involved because she's mated more times, meaning that the males in the low harm treatment or mating to the females from the low harm treatment are doing significantly better and therefore having their fitness altered by female experience as we predicted. So to conclude that, there does indeed to be some good evidence here that experience does influence the behavior of her subsequent mating partners. But one thing I want to point out that it's very difficult to disentangle if these effects are actually due to female plasticity, which is disappointing because I know that was the point of my project, or is it due to male manipulation? So in other words here, it might not necessarily be that these females are making these changes in their behavior to reduce future mating opportunities for males. It could be that the previous females might have experienced more of this exploitative manipulation that we saw in the second project from the males in the high harm treatment. So as a future direction, this is something we're going to be working on. The problem is I've thought about it so much, and it's so hard to disentangle because both of those aspects are so closely integrated that it's just almost impossible to separate them. So I just want to end up with a few other future directions and more broad general discussion. So of course, first I have to conclude my PhD by finishing that fourth aspect that I never had the pleasure of diving into and looking at how female mating experience affects her own fitness. So in other words, can plasticity and the amount of harassment experienced help females reduce the harmful effects of future males? And then from a more bigger picture perspective, I think there's a lot of work to be done, maybe in a postdoc or maybe for someone else's project, to explore how plasticity affects more broad behaviors such as aggression and mood and other types of behaviors, even in species like humans, because it's very clear that plasticity has a huge effect on this variation we see in different behaviors. So why would we not predict that it has an effect on other behaviors as well? So before I finish off, I just want to end things, I guess, in a relatively, helpful way for me and hopefully for everyone else by outlining the 3 most important things that I felt I learned throughout my research, other than the very highly specialized skill of telling the sex of a fruit fly just by looking at its genitals under a microscope. So first of all, the interactions between the sexes, as hopefully you gained from this presentation alone, never mind the entire literature, are very nuanced. And this, I think, is important because, as I mentioned earlier in my talk, it's understanding the evolution of sex and the consequences of reproductive traits are essential for understanding evolution at large, because in sexually reproducing species, evolution can't occur without sex. Second, I think I really understood that when we're thinking about quantifying fitness or evolutionary fitness, how well an individual does, we have to consider many different metrics. So when I was designing my experiments, I was pretty, I guess, narrow-minded, you can say, when I was trying to figure out, like, should we just measure lifespan or measure this very specific timing of offspring production on this day? But the fact of the matter is there are lots of different aspects to what results in an individual being reproductively successful over their entire lifetime. And not to mention that different traits, as we see due to plasticity, might be beneficial or might be adaptive in one environment, but might be completely maladaptive in another. So this is really important when we're thinking about issues that are even important to humans. So things like depression, anxiety, and other types of behaviors that might appear to be maladaptive, but we can't truly understand whether or not they are without putting them into the evolutionary framework and understanding why they evolved in the first place. And then finally, I definitely learned from being in PNB that interdisciplinary integration is absolutely essential. So more broadly, from studying mostly biology literature, I was really, really impressed with how well biologists have been able to integrate aspects of genetics, physiology, neuroscience, ecology into this one big field. And that's why joining PNB I thought was a real treat for me, because it's a relatively new area to me, and we are very lucky to be in this really integrative department. And I just want to take my final words to emphasize the importance of integration and potentially even considering how we can use aspects of animal behavior and biology to bridge the gap between the biological sciences and the more human social-centric sciences. And with that, I would like to thank, firstly, my supervisor, Dr. Ruben Dukas, who has just been absolutely an amazing supervisor. For those of you who know me well, I can be pretty disorganized all over the place. And Ruben has really, really taught me the importance of being very critical and very meticulous throughout the whole scientific process. And I'm confident I'll carry that with me for the rest of my life. So thank you, Ruben, and then also my great lab mates, Andrew, Carling, Tova, and Joshua. My committee members, Dr. Paul Andrews and Dr. Ian Dworkin. And then, of course, the large number, probably longer than this, but these are the independent project students that helped me make this research possible. Without them and funding, it wouldn't be. So lastly, I'd like to thank everyone here for attending my talk, but more broadly for being excellent colleagues over the past 4 years here at Mac. And thank you for listening. I'd be happy to answer any questions now. Yeah, Jim.
Nice talk.
Thank you.
I'm wondering, in your last experiment, when were you, I couldn't really tell whether you were trying to get at the experience that female is having of having multiple males in the trial with her, or whether it was to look at the possible impact of multiple ejaculates that were going to potentially.
Yeah, no, that's a great question. And that's kind of what I was touching upon when I said in my conclusion there, we can't really tell based on my results whether or not it's from the experience or from the multiple ejaculates. But to answer your question, I was trying to get at the aspect of plasticity. That's kind of how I designed my thesis. And as I said, where I'm at right now, I haven't been able to really devise a strong way to disentangle those two components, because the plasticity would be induced by the ejaculates, right? So if we're taking them away, you can get spermless flies. You can get flies that don't transfer ACPs, but then that's an essential part of the mating experience, right? So
One other possible way would be using some normal flies, at least, which I don't know if you can crazy do with genitalia of one male or something, so that
Mhm.
female might be getting only ejaculate from one male or two ejaculates.
Right.
I don't know if that would help with any of this, but
Yeah, no, it's something definitely to consider. So yeah, I'll think about that. Thanks. Yeah, Sigal.
Really nice talk. Thank you.
Thank you.
I wanted to comment on the second question.
Sure.
I guess I would say that I'm really looking forward to your part 4
Mhm.
because there's this kind of overarching kind of feeling in your talk that females are just these passive vessels, and they're absolutely not. And you know that from your third set.
Yeah, that's the whole rationale because I don't think that's true.
I know you
Yeah.
don't. I know you don't. But it's going to be really nice to see that balance.
Mhm.
We don't have enough of that balance
Yeah.
in our field. The second thing is a question, which is, it just struck me as really weird that you take photographs every 2 minutes. A lot of sets might have sets very quickly. So why not take it or live sport?
They
Go on forever.
they will, as far as I know, unless there's like a mechanical complication, they will never mate for less than like 8 to 10 minutes. Yeah, I know it sounds surprising relative to most similar species. They mate for a long time. Yeah. Yeah, 2 minutes is adequate to capture every mating. Yeah.
Thank you.
You're welcome. Thanks. Any more questions? Yeah, Rachel.
I have no background in, I'm just really curious about the harassment
Mhm.
factor, so being in the wild. In a real world situation,
Mhm.
so when there are more competitors, what would that look like? Would having more competitors with the female be ever in a situation where she could not escape?
That is a really good question, and the answer, I think, is actually no. The female can just fly away in nature. And it's actually been a really big criticism of a lot of the sexual conflict research in Drosophila because they've, lab at U of T recently, or Ottawa actually, but that's the Reuven and me crap, did an experiment where they manipulated the size of the enclosure. So they either looked at a big enclosure where the female can fly away or a little vial like I used. And the effects of male harassment are much, much lower in the big cage where the female can just fly away. So there definitely is an unnatural aspect when it comes to that. But with that being said, in nature, the effects of harassment are still going to influence a female's fitness in terms of the amount of time she spends, that she could be spending laying eggs or finding food. So yeah, it is a bit of an unnatural aspect to it, but it's still relevant, yeah. Yeah, Paul.
I just wanted to know if you could comment about latency of mating and whether or not that is always the best measure of fitness, because there can be a trade-off between courtship effort as well, and you can take longer, and I don't know if that actually can produce higher quality offspring.
Yeah, that's a great question. So as far as I know, latency is a standard in the Drosophila literature as a metric of male attractiveness.
In a competitive environment, I can see that firsthand could be very important.
Yeah, even in a no-choice environment. So even when there's only a single male, there's good evidence to show that a male success or the latency, so shorter latency typically correlates with attractive traits. So that's kind of what I was getting at. But with that being said, absolutely, that was kind of the second point I made in my conclusion, that there are other things we might have to consider when it comes to overall fitness. But I guess what I was getting at is I was using latency as a metric for attractiveness. Yeah. Yep.
So, guys, when you're remating the losers and the winners
Mhm.
multiple times, the one experiment, I think, you 24 hours between those two matings, 1 day,
Yes.
and then another one is 3 days. How does that differ if you let, when you're counting paternity,
Mhm.
if you have the individuals that are the males being mated a day apart or 3 days apart, versus if you let a female mate with one male and then immediately swap out and put a new male in and let them mate at approximately the same time? You get a different distribution because you now had one male sperm for 24 hours before the next that sperm is introduced?
Yeah, I would say almost certainly. So one reason why we don't do it right away is because females will not mate multiply within a short period of time. And one of the reasons for that is because one of the pesky accessory proteins that males are transferring are manipulating females to reduce their mating refractory period. So that's why we wait the day. I definitely would expect to see differences though, because the longer the sperm is there, the longer time there is for any transferred proteins to take effect, because those take time to affect the female as well. Yeah. Any more questions? Okay, well, thank you. We are apparently having a wine and cheese, so if you want more questions and wine and cheese, join me there. Thank you.